Hi everyone, and welcome back to the BioStudent project. Today we will be talking about DNA replication, which is the process of producing two copies of a DNA molecule. This is a very important process for cell division. DNA replication occurs during interphase and in the nucleus of a eukaryotic cell. First, DNA replication begins at the origin of replication. Humans have around 100,000 of these spots, which are made up of specific DNA sequences. As a reminder from the last video, DNA has a double helix shape, so it has two strands. Each of the two strands serves as a template for the new DNA we are creating, so in total, 2 new copies of DNA will be made after the process is complete. This is known as the semi-conservative model because each double helix is made up of one old strand and one new strand. The first step of this process is using an enzyme called helicase, which unzips the two strands of DNA by breaking the hydrogen bonds between the nitrogenous base pairs, like A and T or G and C. Enzymes are a very important part of DNA replication. For the most part, you know you are dealing with an enzyme when it ends with the letters A, S, E. Then, another enzyme, primase, goes to an origin of replication and puts down an RNA primer. This primer tells polymerase, the building enzyme, where to start. Polymerase reads the bases from the original strand and places its complement onto the new strand. So, for example, if on the old strand there is an adenosine base, polymerase will build a thymine. There are two ends to a DNA strand, the 3' end and the 5' end. The numbering has to do with the carbon atoms in the DNA backbone. DNA polymerase always starts at the 3' end. But remember, it's building the complementary strand, so if it starts at the 3' end, it will create the 5' end of the new DNA. In DNA replication, there are two strands, the leading strand and the lagging strand. The leading strand is the strand of DNA that is synthesized continuously, while the lagging strand is synthesized discontinuously. The leading strand is built in the 5' to 3' direction towards the replication fork, which is where helicase is unzipping. The lagging strand is synthesized in multiple pieces known as Okazaki fragments, named after the scientist who discovered them. Each of the Okazaki fragments needs a primer to start synthesizing again, and ligase glues them together to create a continuous strand. Topoisomerase, another enzyme, makes sure that DNA does not get tangled by making small cuts in a strand and then sealing them back together. Here is a good diagram of DNA replication. Here at the top you can see the leading strand, which is being synthesized in the 5' to 3' direction in the direction of the replication fork. DNA polymerase is building the new strand and helicase is unzipping the strands. At the bottom is the lagging strand. You can see the pieces, Okazaki fragments, and polymerase is building with the help of an RNA primer. There is also ligase, which will eventually glue the pieces together. And then topoisomerase is also there, cutting small pieces into the strand to relieve tension and prevent entanglement.